feeding competition between 3 cichlids in the Komati system. The reason we were doing this is because I've been working up in the Komati system for a few years, and the one dam within the Komati system had a complete change in the species composition. It's gone from having dominated by Orange-fringed Mozambicus and Komati River, and it's completely changed. It's almost like 99% cocktail granular. Komati River is pretty much extinct in South Africa, and Orange-fringed is amazingly almost extinct in that particular dam. So the reason we're doing this is to sort of try to explain why there's a huge change in composition. I'm not sure feeding competition is the reason, but this is the first attempt at trying to understand this change. Welcome, Phumza.
Morning. Good morning, everyone. Thanks again for coming and listening to my talk. Today, I'm going to talk to you about the feeding traits as predictors of food competition among the cichlid species in the Komati River system. Next. Functional feeding traits of organisms provide valuable insights into predicting food web structures. Because the abilities of predators to capture and consume prey has an underlying functional basis and is restricted by their various morphological features. And then several key functional feeding traits have been identified as strong predictors, such as the gut length, the tooth shape, the mouth width, the mouth orientation, together with the body depth. For example, the gut length, it has something to do with the type of prey that an organism is more likely to consume. For example, larger gut is associated with organisms that feed on prey that takes a longer time to digest, such as the plants and the detritus. And then the tooth shape has something to do with the amount of deformation after chewing. The mouth width has something to do with the size of prey entering the mouth. The mouth orientation is related to type of feeding. The body depth is associated with swimming, for example. A narrow body depth is associated with organisms that are fast swimmers, more likely to chase their prey for feeding. And the feeding morphological structures can be used to predict food competition between species. And hence, in my study, I have looked at the Orange-fringed Mozambicus, which is vulnerable to extinction, the Komati River, which is least concern, and the Redbreast, which is endangered. These species are all occurring in the Komati River system, and based on the literature, if organisms are similar, they are more likely to compete for food resources, and that may have a negative impact in their numbers. And also another thing is that there is a noticeable decline in the numbers of Orange-fringed Mozambicus and Redbreast in the river system. And also another interesting thing is that though the numbers of Redbreast have been noticed to be declining, its type locality is in Komati River, which is also part of that river system. And then in my study, I've looked at the diet together with the feeding morphology of the study species. To do this, specimens were collected in those 3 dams illustrated there. And then the specimens I've requested from them from the SAIAB collection. And in total, I used just 10 individuals per species. And then these are their size ranges. These are the size ranges of specimens that I used. And the specimens were collected in winter of 2012. For my methods, morphological measurements were taken following the procedures outlined by Nagekelke et al. 2018, whereby the diagram there, it shows the morphological measurements that I took in my fish, and I used the Orange-fringed Mozambique as an example. The measurements that I took are those that are responsible for the prey location, the prey chasing, the prey capturing, the chewing process, together with the digestion. And then after that, a PCA was performed on standardized data to compare the overall morphology of the three study species. And then after that, a food fish model which was developed by Siping and Nakekelke, 2001, was used to quantify the ability of functional food trays to feed on different food types, such as the larvae, the chironomids, the detritus, the phytoplankton, the plants. It's many food types. We're going to get to a slide where they will be illustrated. This model, it tells us that based on the morphological, based on the size measurements that we took from each fish, what are the chances are that this fish is going to feed on mollusks? What are the chances it's going to feed on chironomids, and so on and so on? Next. Dietary analysis were conducted on the gut contents following the procedures outlined by Highslope 1980, whereby the prey abundance, the frequency of occurrence, and the percentage of volume was determined, and the index of relative importance was determined using the equation. And then to compare the diet between the three study species, I used the percentage index of relative importance. And this diagram here, it shows some of the gut contents that I found out from the Coptodon rendali. And then for my results and discussion, PCA ordination represented 42.7% of total variation, indicating a difference in the trophic morphology. If you can see from this diagram here, the Orange-fringed Mozambique together with the Coptodon rendali are sitting closely together. And they are slightly overlapping. Chances are these organisms are more likely to feed on similar prey resources than the Chytia brevis, which is sitting separately there on its own. And then the directions and sizes of our loadings tells us that the Orange-fringed Mozambique together with Coptodon rendali were characterized by long guts and body depth, and meaning that this one is it will have more of plants and detritus in this diet, both of these ones, and also will the larger body depth tells us that it is less likely that it chases prey for feeding. And then looking at the Chytia brevis, Chytia brevis is characterized by large operculum area, large head length, oral gap axis is large, lower jaw length is large, and meaning that these characteristics are for organisms that are feeding in the water column. And then these are my trophic profile results. Here we have different food types that I spoke about on my methods. They are listed here. And then we have different three rows of circles, which represents our species. And we have the circles with different colors. The green is for positive correlation, and then the maroon is for negative correlation. And the bigger the green or the bigger the maroon, it means that the bigger is the correlation. Looking at the first one, it has a strong positive correlation to zooplankton together with larvae, and it's more likely to catch fish by chasing when feeding. And then the Coptodon rendali, on the other hand, is mostly correlated to all the tested food types except for fish and insects. And then on the other hand, Oreochromis mozambicus has a positive correlation to plants, a strong positive correlation to plants, fish, insects, detritus, and algae, and has a negative correlation to zooplankton. And then this diagram here, it shows us the results that are found from the gut contents of my study species. And then when everything that I've highlighted here in green, it's matching with the previous slide. And the maroon is also matching with the previous slide. If you can see from this graph here, the Chytia brevis has a positive correlation, as predicted from the previous slide, that it's more likely that it's gonna feed on larvae. There's a presence of there's a high consumption of chironomids had a high contribution in the diets, and also it had zooplankton, as also predicted from the previous slide, and there was also the presence of amphipods that was also noticed. And also it's predicted from the previous slide that this one is more likely to be feeding in the water column. There's an absence of as predicted from the previous slides, that this one is not gonna feed more on hard prey. Algae and plants are absent in their diet, and also from the previous slide, it was predicted that it won't feed on detritus, and if you can see from here, mud and sand are absent in their diet, whereas they are recorded on the other species. And then moving on to Coptodon rendali. Coptodon rendali, as predicted also from the previous slide, that it will have high consumption of detritus. We can see that there's a presence of sand and ostracods in its diet. And also there is a presence of algae and zooplankton because of the presence of Cadocera and the calanoida. And then Oreochromis mozambicus, on the other hand, is more feeding on mud on their gut contents. There was high volume of mud and sand, together with there was also a presence of algae and plants. And I wouldn't say that this plants only feeds on chironomids. That is why we're planning to go and sample more specimens so that I can analyze, maybe if I can have maybe about 20 specimens for diet, that will give me a better results. And then if this diagram is this table is telling us that the Oreochromis mozambicus and Coptodon rendali have overlapping diet as predicted from the previous slides. And it's clear that the Chytia brevis has a different diet. Also, the presence of algae, plants, and detritus is common in the diet of Coptodon rendali and Oreochromis mozambicus, and this was observed in a study by Zengeya et al. 2011 in the Limpopo River. In conclusion, feeding morphology traits are good predictors of food competition between the study species. Oreochromis mozambicus and the Coptodon rendali, they are benthic feeders. I wouldn't say that the competition for food resources does contribute to the decline of Chytia brevis, because this species, it had a different diet with its closely related with the other two species. Whereas I would say that the decline of Oreochromis mozambicus could be as a result of Coptodon rendali, since they are competing for food resources. However, future studies can look at the impact of other alien invasive species that are present in the diet, that are also present in the river system. Thank you.
Any questions?
Thanks a lot. It's a lot of hours under the microscope of sorting out gaps and analysis. And I wanted to ask you, instead of jumping onto the invasive species, perhaps if you still want to validate this analysis, the methodology, wouldn't you want to consider maybe looking at the diet over different periods of the year? Because maybe you may have some overlapping or not, or success of your validation for this specific thing, but during other periods of the year, the animals, I don't know, they may be less choosy, or they, I don't know, do they have to, can they adjust their diet depending on the season?
Okay, consider that, Francesca.
We were constrained with the amount of material we had in the collection. So, yeah.
But if we check that when the specimens that we looked at were collected, were they collected at the same period of time?
And yeah, it was winter of 2012, all of them.
In the
Yeah, all of them.
Are these generally like adult, juveniles, all mixed?
The reason we want to increase the want to do another sampling now, it's because we want to catch more of the larger size individuals. We didn't catch enough numbers for the large sizes.
I think that it's definitely good to consider that. Bearing in mind that sometimes the juveniles intake and even diet patterns will be actually quite different. Yeah. Also, you mentioned that you only want to take at least 20. Forgive me for my ignorance. I don't know how big the dam is. Is there a need to do more sites? Like, what's the behavior like? Are they, stay in crevices or something? And
Sorry?
do they stay in crevices? Do they hide and stuff? So is 20 representative of the whole dam?
No, the 20 is for my sample size. I'm trying to increase it, more especially for the dietary analysis, because it's not enough for the diet, 10 individuals.
I see when you were comparing the three species, Oreochromis was the only specimen, is the only species that feeds on mud. Is there any reason why is it feeding, or is it unavailability of food, or they are just opportunities, they take anything that is available?
They fit in the bottom, maybe that's why mud, maybe that's why mud is present in their diet.
I think the feeding on algae is incidental. You have a question?
There's someone online.
Oh, sorry. Okay. Mananga.
The chat seems to have.
Right. So you're gonna-
Buddy had his hand raised.
Can you enable the mic?
So this is Craig. I'm not sure where Craig is right now. Thanks for the great talk. You mentioned non-natives as another possible cause of decline, but what other drivers may be present?
Maybe fishing also.
There is fishing going on in there, and there are alien Micropterus floridians in the dam. Yeah, they do. Yes. And so the changes happen since the bass were put in. So that's why we do think that it's
So this bass was intentionally put in for,
Yes.
for recreational. Okay.
all over the country, bass are put into dams.
And then it's like an open season or something.
Yes. Yes.
All the time.
Bass fishermen seem to have this philosophy where there's an open body of water that doesn't have bass in it.
Oh, okay.
It's illegal, but gets done almost instantly. Some dams are filled with bass before they've actually filled up. The Guga Dam, which is one of the Swaziland dams, before it actually filled up, they had bass in. Construction, still filling. And this change has happened since then. So sorry, Ule, we compared specimens with other river systems and tried to see evidence of morphological plasticity. So again, we're coming down to the numbers of samples we have available to chop.
No.
Because it's just struggling sampling.
No, Ule, I haven't compared with specimens from other river systems.
So you had a.
In your introduction, you mentioned that two of your species, one is vulnerable and one is endangered, and you are planning to increase your sample size for the next level of your study. Will that not have an impact on the population of those two species, or you omitting those two species going forward?
Usually when people, when people are describing diet, they collect numbers like 30 to 40. That's why I'm trying to minimize my sample size to 20, because even 20, I wouldn't say it's enough to explain the diet.
The Orange-fringed is listed as vulnerable based on possible hybridization with Orange-fringed. That's a widespread assessment. But I mean, where it occurs, it's pretty common. Orange-fringed populations all over South Africa, there are lots of healthy populations. So collecting those isn't a problem. Brevis is almost extinct in South Africa. But we'd like to use in Swaziland. There's a barrier weir as you go into Swaziland, and above that weir there's a healthy population of Brevis. There are hundreds of them. So that's where we'll be collecting the next round. We good. That is. Last question.
Sorry?
Sorry?
No, I don't know what the Pule raises hands again.
Yeah, no, I'm just following up on the question that I good morning, everyone. I'm just trying to follow up on the question that I posed on the chat, the box there. So it's just a comment, Phumza. Thanks for the presentations. Thanks for presentations. It's quite refreshing to see studies being done on ecomorphological traits. It's quite close to me. So I just wanted to make a comment with regards to morphological plasticity, right? For example, I think it will be very interesting to actually try to compare your specimens from the Komati and other river systems and try to see if maybe there was a change in morphological feeding traits of those species that you studied, because I'm thinking now, I think with for one of his theses, I couldn't remember if it was master's or PhD, where he was looking at the labios. I think it was umbratas, actually. I think it was labio umbratas. So he actually found that the populations of the Orange River system had different morphological in terms of morphology. They had a different morphological they had different morphology with the populations of the Great Fish River. So there were differences between morphology in terms of the mouth. I think it was the mouth when he found that, and so I think it will be quite interesting to see if the similar pattern is observed as well with your species there. If maybe, for example, your Oreochromas change their morphology, try to alleviate the competition or ease the competition. So I think maybe, but I don't know, like you said as well, those species are endangered. So the issue again becomes the issue of getting enough sample size. But I think it will be interesting to try to compare those and try to see. Yeah, that's just that.
Nice, good comment, Pule. I do agree with you, because the type of prey exposure, it may have an impact in the feeding morphology of the organisms. I've noted your comment.
Any other questions? Are we done? Vanessa?
Any way of getting rid of the bats? Or does that mean you're gonna put them back in again?
Any way of getting rid of the bus?
One of the things that Albert's going to be exploring is the possibility of introducing special hybrids that will interbreed with bats that will produce sterile offspring. So that's something that they're toying with, but it's far away from actually happening.
And takes time too.
Yeah.
You know.
I mean, one of the things that Nature Conservation in Mpumalanga are doing, I know they're doing it in Loskop Dam, is that they go in the spring and I think they spearfish bass on nets, which also means that the juveniles that have been spawned that season get taken out very quickly by the fish. And they seem to think that's a great way. I mean, to me, that's just a never-ending spearfishing holiday for somebody. But yeah, I think the way Albert's talking about it is the way to go. Oh, yeah, it's a problem, a huge problem. Thank you, Phumza.
Thanks.
Very, very much. Oh, no, one more question.
Do you have like a specific reason of looking at the diet of these species, only the diet of these species?
Only these ones.
Yes.
That's because I've said that some of them are declining in numbers. And food competition is one of the things that may contribute to the decline of fishes when, say, another one is over competing the other one.
So I'm not a fish person, so are they edible? Is that edible? Fish? So do you perhaps like look at the fishing?
I didn't look at it for this particular study, but another study that is looking at that may also.
Thank you very much.